Okay, last leg of the journey for this week. We're going to talk about complex polygenic inheritance. Most disorders that you will encounter working clinically, unless you work as a genetics nurse, are going to fall under this umbrella. And you'll see the leading causes of death and disability all around the world are considered, most of them are complex polygenic disorders. So monogenic disorders occur, a single gene alters a protein that's important for the body to function. We've talked about them extensively. But most disorders don't involve a single protein. Most involve a complex system of tissues, cells, hormones, enzymes, neurotransmitters, all working together. So many genes contribute to the overall function of that system. For example, it's really important for the body to maintain a steady supply of glucose, but not to allow a hyperglycemic state. Without glucose, your brain and most of your tissues cannot function. It's the only fuel the brain can use. So the body prioritizes this, and it has many different proteins that make sure that we have enough glucose and just the right amount in our bodies. For example, we have insulin and we have glycogen storage enzymes. We have all kinds of mechanisms for this. So in order for the disease to develop, there are all these fail-safes in the system. You need several faulty genes, and you need some dietary and lifestyle factors. Now, these genes are actually fairly common, these alleles that predispose to type 2 diabetes. And there are also epigenetic factors, things that if your ancestors experienced famine or starvation, it switches on genes that promote obesity. It's a protective thing, but it is passed on through the generations. And we're going to watch some videos during this semester that kind of underscore that. So it's important. You can't predict risk from a simple test for a gene or from a Punnett square. You need to learn how to get a careful family history that goes back at least 3 generations, and you have to know how to interpret that. In the future, genetic testing may be more helpful. At this point, not all of the genes implicated in obesity, diabetes, hypertension, atherosclerosis have been identified. So it gets a little bit trickier to use it, but they are coming closer. Do not trust companies like 23andMe or My Ancestry or any of those places that tell you they're going to give you a report based on your genes from the little kit that you swab and send in for $79 or whatever. Do not trust that report. The science just isn't there yet. We can see some things and not others. It will evolve in your lifetime, and hopefully at some point we'll be able to offer people individualized health plans. So some examples, a lot of birth defects like cleft lip and palate, neural tube defects, congenital heart defects. These have a genetic sensitivity to teratogens. So if they're exposed to that teratogen, or if they're deprived of folic acid, we'll say, individuals with that allele or those alleles are more likely to have these disorders. Autism spectrum disorder, very complex polygenic. They have identified several places in the genome that are implicated. And because brain function is so complex and relies on so many different processes, it's not surprising that things like bipolar, schizophrenia, attention deficit disorder, substance use disorder, alcoholism, all of these things have polygenic inheritance. And they have environmental triggers. Interestingly enough, schizophrenia is considered one of the most heritable diseases, meaning that having a close relative greatly increases your risk above the general population. However, in studies of identical twins, people with the same exact genome, if you have a sibling with schizophrenia, you are only at 50% risk, 50-50 chance that you will too have schizophrenia. So it's worth investigating what are the environmental factors in some of these disorders, and what are the genes, and how do we mitigate risk for people, because those things are devastating to have. Alzheimer's disease, several genes implicated in that. We talk about APOE4, but there's also the SPP gene that's located on the 21st chromosome, and that codes for protein that is also a precursor to the amyloid plaques that build up in the brain, which is why people with Down syndrome who have an extra chromosome 21, who make 33% more of that protein, usually end up with Alzheimer's at some point in their lives. It's an overwhelmingly large number. Obesity, atherosclerosis, diabetes, hypertension, those things all go together because metabolism is a complex process governed by many, many different genes and proteins and your autoimmune disorders, asthma, epilepsy. Oh, no, epilepsy is not autoimmune, but lupus, rheumatoid arthritis. There are multiple genes that are responsible for the inflammatory response, and having variations of those alleles that make you more predisposed to them, and also having environmental triggers can really influence the course of that disease. Congenital heart defects we already talked about. So those are some examples, and this is why it's so important that in this course you learn how to take a family history and do a pedigree, because it's still the best tool we have at the moment. All right, we are going to wrap up with Ploidy. It is not very extensive. A couple of the concepts are tricky. I'll walk you through them. If you have questions, please reach out to me, either through your responses in the Edpuzzle or through email or any other method. Come to my office hours and I'll explain them to you. So ploidy is just the actual number of chromosomes in the nucleus of the cell. You can be euploid, the prefix EU or U, meaning good. You have the good number. In a human, not to beat a dead horse, it's 46 in a somatic cell, 23 in a sex cell. Or you can be aneuploid, and that prefix an means not. So not the good number. You have the wrong number of chromosomes in the cell, and there was a mistake in cell division. Instead of a parent cell equally dividing its material, one chromosome got stuck to the other half of the pair, and now you have two chromosomes in a sex cell where you shouldn't. That's when it happens in the germline. But it can also occur in a somatic cell. So as cancer evolves, you get exposed to carcinogens, DNA gets damaged, it reproduces in a chaotic pattern, and you usually get aneuploidy. And that tells you cancer is advanced. Karyotype, that's a picture of a karyotype. It's actually not a human one because there's not 23 pairs. There are 29 plus the sex chromosomes. I don't know what species that is, but it isn't human. But it's just a snapshot. It's a picture. They basically extract the nucleus of the cell. They look for, the phase where the cell is dividing, the chromosomes are dividing, and they take a picture and then they stain it, or they stain it and then they take a picture. It does give limited information. It's not going to tell you much about the genome, which genes are on the chromosomes, but it does show the structure and number, and if there's something grossly abnormal, you'll see it. When you get a karyotype, it's reported as the number, the ploidy, and then it's followed by the type of sex chromosome. So, for example, normal human assigned female at birth is 46XX. That person has 46 chromosomes, and the sex chromosomes are XX. Normal assigned male at birth is 46XY. Someone with an extra X chromosome, just means, for example, would be reported as 47XXX or 47XXY. So you would know that that person was aneuploidy and you would know what they had. All right, types of aneuploidy in sex cells. An ovum or a sperm divides in a process called meiosis. That is like basic biology, and you don't really need to recall it for this. Just know that females are more likely to have aneuploidy in the sex cells. It can happen in males, but it's less common because men continually produce sperm, but women are born with all the eggs they will ever have, and those eggs age. The cell quality declines. The telomeres get shorter on the chromosomes. DNA is more likely to be damaged, and chromosomes can get stuck together. So one daughter cell will have an extra chromosome, and the other one has fewer than needed. If that happens during the ovarian cycle and the egg is released and penetrated by a sperm, the result will be aneuploidy in the offspring. So the most common type that we see is trisomy, where the offspring has 3 copies of a chromosome instead of 2. You're familiar with trisomy 21, which is Down syndrome. There are only a few of these that are compatible with life. Monosomy, the offspring is missing 1 chromosome in each pair, so they've got a 45, either XX, XY, or XO. There's only 1 monosomy that's compatible with life, with human development. That is Turner syndrome, and the missing chromosome is an X chromosome. And because of X inactivation, there are some features associated with, but people live relatively normal lives. Other types of monosomy are not compatible with life. You need both chromosomes in most pairs. Polyploidy is a little bit different. This is when the parent cell, instead of having one extra chromosome, has an extra copy of every chromosome. So you can have this with one parent cell, one gamete, so one ovum or one sperm, or you can have it in both. And the resulting offspring or the resulting product of conception will have 69 or 92, totally incompatible with life, but it also creates conditions that are conducive to cancer in the mom. Most of these mistakes that happen during cell division end in early pregnancy loss because most chromosomes carry so much material that having an extra copy creates absolute chaos, and so the human can't develop any further. Sex chromosome aneuploidies are more likely to survive. Many of them are ending in early pregnancy loss. It just depends on which alleles are actually on those actual chromosomes and what they code for and whether or not that's going to be a huge problem. But because of X inactivation, you might see people living actually normal lives with sex chromosome aneuploidies. Trisomy 21, so the chromosome 21 is the smallest, not the smallest, the 23rd pair it's arranged in size, but it has the fewest genes, and the genes are not considered as essential to life. That's why you see people with trisomy 21 who are born and live to be 40, 50, 60 years old. They usually don't live much longer than that. It depends on which actual alleles are on the actual extra copy and how much extra proteins you're going to get from that. And that will impact the severity of the intellectual disability. But yeah, those are types of aneuploidy sex cells. We're going to talk about mosaicism briefly. Mosaicism happens. Both parent cells are normal, okay? Some cells in the body develop aneuploidy early in development. So when they're rapidly dividing, whether it's in that undifferentiated stem cell stage or whether it's a little later in embryonic period, cells are rapidly developing and they're more likely to make mistakes. It doesn't happen frequently, but it can happen. So the parent gametes were normal. And at some point after conception, during cell division, one cell gets that extra chromosome. All the cells that are descended from those aneuploid cells will also be aneuploid. They will look like the parent cell. The other cells in the body will be euploid. So it depends on how early it happened because that's going to determine what percentage. But the individual will develop some features of the disease. But depending on which type of cells the aneuploid cells eventually become, if they become a type of cell that really doesn't have to code for the proteins in most of those genes, how many cells there are, features are usually less severe. And you can have people who have cells in their body that are trisomy 18, which is usually lethal, and they're walking around and they're normal. I will give you case studies on that. But some people, there's one that fits really well with ethics for next 2 weeks from now. Some people are asymptomatic. They're only diagnosed either through prenatal testing when their mothers have amniocentesis or CBS or NIPS testing, or if there's some other reason that they need genetic testing. Maybe they're pregnant and they want to see their own karyotype. But there's a few people out there. I have some great videos on that. We'll get to it later. But that's the concept of mosaicism. So you may have somebody with a mosaicism of trisomy 21, and they have some features of Down syndrome, but a lot of they function at a much higher level, and they have fewer incidents of the diseases associated with Down syndrome. Okay, now we're going to get to the trickiest part. I do not want you to rack your brains over translocations. There is a lot of content in this week. I had to kind of pack it in to give you basis for other things. It does get easier. A translocation is what happens when genetic material gets misplaced on the wrong chromosome. It happens. There are two types. There's balanced and unbalanced. We're only going to really worry about one because we're just talking about ploidy right now. Balanced translocation, all the necessary genes are present, but some of them are on the wrong chromosome. So the phenotype is still normal because all the genes are still doing all their jobs. They're just kind of in the wrong place. There's one type of balanced translocation called a Robertsonian translocation. Please do not go crazy memorizing this, but try to picture it, and I have a video that'll help. The affected individual will have a karyotype that reads 45XX or 45XY because two chromosomes are stuck together. It only happens in certain chromosomes. These are acros. Don't worry about it. But 13, 14, 15, 21, 22. Now, trisomy 13 is sometimes compatible with life, and so is trisomy 21. These others, 14, 15, and 22, are not compatible with life. But when this individual with the balanced Robertsonian translocation has these stuck together chromosomes and their egg or sperm cells divide and become mature sperm or ovum or ova, some of those cells will have a double dose of a chromosome because they will reproduce. Let's say it's 14 and 21 are stuck together in one pair, and in the other pair, 14 and 21 are standalone chromosomes. What will happen is they will get a double dose of either 14 or 21. I know that sounds confusing. I don't know how to make it not confusing except to play this video.
A Robertsonian translocation carrier describes a situation where someone has 45 chromosomes instead of 46. Robertsonian translocation only occur between the acrocentric chromosomes. These are chromosomes 13, 14, 15, 21, and 22. What happens is that two acrocentric chromosomes get stuck together, making one large chromosome. The commonest chromosomes to become stuck together are chromosomes 13 and 14, and the next most common are chromosomes 14 and 21. Other possible translocations occur much less frequently. A Robertsonian translocation carrier is healthy. However, problems can arise should the Robertsonian carrier wish to have children. To become a parent, you have to make a mature egg or sperm. Each parent only hands on half their chromosomes into the egg or sperm. Normally, each pair of chromosomes come together, exchange genetic material, known as crossing over, prior to dividing. For Robertsonian translocation carriers, this process is more complicated. In this example, Carl is a translocation carrier. He has one chromosome 13 stuck to chromosome 14. He also has another normal standalone chromosome 13 and 14. If he hands on the normal chromosome 13 and the normal chromosome 14, then the child will be healthy and have normal chromosomes. If he hands on the translocated chromosome on its own, that is the 13 and 14 stuck together, his child will be a healthy translocation carrier like himself. However, if Carl hands on the translocated chromosome together with one of the standalone chromosomes 13 or 14, the baby would have a whole extra chromosome 13 or 14, as Carl's partner will hand on a standalone chromosome 13 and 14 too. The baby will end up with 3 copies of either 13 or 14. This is known as trisomy chromosome 13 or 14. Babies with trisomy 14 will miscarry. Babies with trisomy 13, known as Patau syndrome, miscarry, although some may survive to birth. The condition is not compatible with life. If Carl carried a translocation involving chromosome 14 and 21, he would have a risk of trisomy 14 or trisomy 21, known as Down syndrome. The risk of having a baby with Down syndrome is higher if the woman, example Carl's sister, carries the translocation. Translocation families can also...
All right, so what I want you to get from that, please don't make yourself crazy memorizing Robertsonian translocations. But if you are working in healthcare or you are friends or family with somebody and they have chronic miscarriages and there's people with Down syndrome or trisomy 18 in the family, you're going to recommend that those people get genetic testing to see if they're translocation carriers. And that's as deep as I would go on a test question, because that is how I would expect you to use that information. All right, so I know that this week had a lot of material. It does get a little less dense, and then it gets a little more familiar when we talk about the actual disorders, but hang with it. And I'll try and break it down for you as easily as possible. Again, I'm here for you anytime you have any questions.